Hello and welcome. Thank you for joining us for today's webinar entitled Early Life Development of Thymic Microbiota-Specific T cells. My name is Jeff Jensen and I will be acting as your host for today's event. First off, we want to thank BioLegend for sponsoring the event. The wonderful folks at BioLegend are always working to bring you fantastic content and exceptional products and services. They have some of the best antibody products available anywhere in the industry, and their website is chock full of useful articles, web tools, and much, much more. If you haven't already seen the content library on their website, you definitely should head over to biolegend.com and see how the terrific information that they've put together can help you and your research. This webinar today is being hosted by FluoroFinder. If you frequently use multi-parameter flow cytometry in your research, then you should get yourself familiar with FluoroFinder, the gold standard of online panel design tools. More information can be found at fluorofinder.com. As a reminder, today's webinar is a live event. There will be a question and answer period at the end, but please feel free to ask questions during the presentation by using the GoToWebinar control panel located on the right side of your screen. The webinar is being recorded, and a link to the recording will be sent out to all registrants. Our presenter today will be Dr. Daniel Zegarra-Ruiz. Dr. Zegarra-Ruiz was born and raised in Lima, Peru, where he obtained his Bachelor of Science in Pharmacy and Biochemistry. He holds Master of Science and Master of Philosophy degrees in Immunology and received his PhD in Immunobiology from Yale University. For his doctoral dissertation, he focused on elucidating the crosstalk between diet, gut microbiota, and autoimmunity, topics that he taught at the Young Global Scholars Program in New Haven and in Beijing. He currently is a research fellow at Memorial Sloan Kettering Cancer Center, investigating host microbe interactions and their effects on cancer and autoimmunity. Dr. Zegarra-Ruiz has asked me to call him Daniel today, so if you're ready, Daniel, I will go ahead and pass you control of the screen so you can start your presentation.
Perfect. Let's do it.
And you should get the screen control right about now.
Okay. Let me know if you can see my screen.
It's perfect.
Okay, great. So we can begin. So thank you very much for the invitation and the opportunity to let me show my research that I've been doing in the past few years. So this research about early life development of thymic microbiota-specific T cells was done at Baylor College of Medicine. It began there and was finished at Memorial Sloan Kettering Cancer Center under the supervision of Dr. Deal. And this was awarded also the Lefrançois-BioLegend Memorial Award for mucosal immunology. And it's going to be interesting to know what other effects does the gut microbes have in the host. So we're going to go straight into it. So we have to remember that we are colonized by trillions of bacteria that constantly modulate how we interact with the world and how we can handle disease. So this bacteria is involved in multiple homeostatic processes. It is important to mention that colonization by this bacteria is important for a proper immune development, for good metabolism, nutrition, and of course is relevant for proper tolerance against other microbiota as well. However, uncontrolled outgrowth of bacteria and different metabolites is influencing disease in a causal, not only correlation manner. For example, we know bacteria are able to exacerbate and even trigger inflammation in the gut, affecting IBD. And also it has been shown to affect autoimmunity progression, for example, in lupus or other autoimmune diseases. Since we are colonized by trillions of bacteria, we need to understand that this bacteria is still trying to infect us. So we need to have different ways in which we can control the proper growth of this bacteria and colonization and distribution in the gut. So for this, we have multiple ways in how we're controlling this bacteria. One is a physical way in which basically mucus is secreted by goblet cells in the intestine. And this mucus is a physical barrier in which allows the host to have a separation to the bacteria that is colonizing in the gut. In addition, we have this secretion of antimicrobial peptides by panel cells, such as alpha defensins and REX3 gamma, that is going to target bacteria and control their growth. In addition, we have a humoral response by the host, for example, IgA secretion by plasma cells in the gut that is going to coat bacteria, and also prime immune responses to control their colonization in the gut. However, and as I mentioned before, these bacteria that is composed in the gut microbiota are constantly modulating immune responses as well. For example, there's a classic inducer of Th17 responses, which are the segmented filamentous bacteria, which in germ-free mice and in SPF mice is known to induce Th17 responses. On the contrary, bacteria from the class Clostridia are known to induce regulatory T cells, which are actually promote tolerance, and other metabolites such as PSA from Bacteroides fragilis are known also to induce these regulatory T cells. But these are just a few examples. However, multiple bacteria-specific responses are shown or have been found in the gut. Like I mentioned before, segmented filamentous bacteria, or SFB, is known to induce Th17 responses. Bacteria from the class Clostridia and also B. fragilis are known to induce regulatory T cells. And Helicobacter hepaticus, as well as E. coli, are known to induce bacteria-specific Th1 responses. Another recent example is with Akkermansia mucinifila that is known to induce follicular helper T cells. So we know that these microbiota-specific T cells exist and are found in the gut, in mucosal cavities. However, there's one important question that remains. How are these microbiota-specific T cells generated? So to understand or to try to answer this question, we need to realize that most of the T cell selection process happens in the thymus, far away from the gut. And this process, which consists of positive and negative selections, is going to basically trigger the development of double positive thymocytes, so basically CD4, CD8 positive, double positive thymocytes, into self-tolerant CD4 single positive and CD8 single positive T cells, which are then going to be exported from the thymus into the periphery. However, this explains only how we're avoiding basically autoimmunity with this process of negative selection. But we don't know how the gut microbes are influencing this process to basically enrich or expand the repertoire that we have of microbiota-specific T cells. We don't know if this process is even happening at all. So for this, we began our research with the question, can we find microbiota-specific T cells in the thymus? We know that colonization with E. coli and with SFB, mainly with SFB, because we can detect these bacteria or these T cells specific against SFB with the help of tetramers. We know that these T cells exist in mucosal areas, such as the gut. However, we decided to find out if we could find these microbiota-specific T cells in the thymus as well. For this, we colonized SFB-free mice with SFB at weaning and at 12 weeks of age. Weaning is basically 3 weeks of age, and we're considering adult mice that are over or around 12 weeks of age. We colonized these mice for 2 weeks, and then we collected the thymus. And using tetramer enrichment, so basically the tetramer is going to help us detect these SFB-specific T cells in this tissue. We're going to do a dual tetramer staining of these cells and then enrich them through a magnetic process in which we're magnetically enriching these bacteria-specific T cells from the thymus. And then we are performing flow cytometry to identify these dual stain tetramer positive T cells or SFB-specific T cells. What we find in the thymus of these mice is that only in mice colonized at weaning, so in young mice, we see the induction of these SFB-specific T cells after 2 weeks of colonization. However, in adult mice, so mice that are around 12 weeks of age, we don't see any induction. As you can see here. Then we went on and analyzed in detail exactly what these tetramer positive cells or SFB specific T cells were in the thymus. Were they Th17, like what we find in the gut, or were they something else? Interestingly, we found that these tetramer positive cells were not ROR gamma positive, so basically they were not Th17 positive cells. They were not Tregs, as they were FOXP3 negative and CD25 negative. And I'm not showing it here, but they were also TBET negative. These cells were also TCR positive and CD24 negative, and also CD69 mid and low. All of this suggests that these cells were actually naive and not coming back from the periphery as mature T cells or as mature recirculating T cells. To test this, what we did was colonize with SFB, not B6 mice, but RAG-GFP mice, which is going to allow us basically to track if these tetramer positive cells have been recently developed or if they are recirculating from the periphery. And what we found is that these SFB specific T cells from the thymus were most of them RAG-GFP positive and CD73 negative. This means that these cells were not coming from the periphery and were actually recently generated in the thymus. To further dissect what exactly was different between these SFB-specific T cells in the thymus compared to all the other T cells we find, what we did is RNA-seq of these sorted tetramer positive cells and compare it to SFB non-specific cells. So first, we compared our SFB specific cell population versus our SFB non-specific SFB population. And I'm gonna show it here. So basically, this is our SFB specific population, and this is our SFB non-specific population. All of this from the thymus. I will compare these populations with a recently published study that showed different signatures according to the developmental stage of T cells in the thymus. And we can see here clearly that both our SFB specific and non-specific CD4 T cells clustered nicely with semi-mature and mature CD4 single positive cells. And they are basically transcriptionally different than immature CD4 single positive cells, intermediate cells, or signal and non-signal double positive cells, which suggests that these cells are clustering nicely with mature CD4 T cells, which are ready to be exported into the periphery. In addition, we compared the basically changes in gene expression with RNA-seq between our SFB non-specific and our SFB specific CD4 single positive cells, and we found that in our SFB specific CD4 single positive cells, there was an enriched in genes that are related to proliferation, to signal transduction, transcription and translation, T cell signaling, and it was basically suppression in genes related to pathways involved in protein degradation. So all of this suggests that these cells are proliferating, have been basically interacting with TCR signaling, and these are ready to leave the thymus. Interestingly, and to assess if this was a specific process or if this was happening randomly in the thymus, we analyzed the thymic subsets, so total CD4 single positive, total CD8 single positive, and total double positive after SFB colonization in young and in adult mice, and we basically found no differences in numbers, or I'm not showing it here, or in maturity of these cells, which suggests that this is a process specific for SFB specific T cells, and it's not changing all of the thymic immune landscape. So since we are able to find these SFB-specific T cells in the intestine of mice colonized with SFB, we then went on and tried to understand if there was any difference in the amount of cells found in the gut or in the periphery in mice colonized at weaning versus mice colonized at 12 weeks of age. And interestingly, we found that mice colonized with SFB at weaning basically exhibited no induction of SFB-specific T cells in the periphery in MLN or ileum, with their frequencies or numbers. However, and that's 2 weeks after colonization. But in adult mice, so in mice colonized at 12 weeks of age, 2 weeks after colonization, we see an enrichment of these SFB specific T cells in MLN and ileum. So then the question is, okay, so now we are colonizing these young mice at weaning with SFB, and we see SFB specific T cells in the thymus 2 weeks after colonization. Are we ever gonna see them in the periphery? And for this, we colonize again SFB with SFB, our B6 mice at weaning. However, instead of waiting 2 weeks, we waited for 4 weeks after colonization. And then we assess for SFB-specific T cells in the periphery. Interestingly, we see that compared to mice colonized at 2 weeks, mice colonized for 4 weeks actually exhibited an enrichment of these SFB-specific T cells in MLN, in the spleen, ileum, and colon. So basically all around the periphery. Interestingly, we also find these SFB-specific T cells in the thymus, just as mice colonized for 2 weeks. However, when we do this assay in RAP-GFP mice to know if these are still being produced in the thymus or if they are recirculating mature T cells coming from the periphery, we find that unlike mice colonized for 2 weeks, mice colonized for 4 weeks actually were mostly RAP negative or CD73 positive, suggesting that these cells were actually recirculating and not being constantly generated at 4 weeks post-colonization. To test if microbiota as a whole had an effect in this process, what we did was deplete gut microbes with antibiotics for 2 weeks. So we gave antibiotics to mice at weaning, and then after 2 weeks, we collected the thymus, and we analyzed basically total thymic subsets. And we found that depletion of the gut microbiota actually decreased total thymic cellularity, and also numbers for CD4 and CD8 single positive cells, and also total numbers of regulatory T cells in the thymus, suggesting that the microbiota actually had an effect in this process as well as a whole. To test that this decrease was not due to antibiotic toxicity that was just shrinking the thymus and just basically killing our cells, we performed the same assay in germ-free mice. So basically, we provide them with antibiotics, but there was nothing to deplete, it was just antibiotics. And we found that unlike SPF mice, germ-free mice actually had no change in the numbers of total thymic cellularity or in the thymic subsets, suggesting that this effect was in fact driven by the microbiota. Good. So we know that we're able to identify microbiota-specific T cells in the thymus, and we've seen this with SFB. However, the question now remains, if we find these SFB-specific T cells in the thymus, are we able to see or to find bacterial antigens as well? So for this, using a similar approach as before, we colonize SFB-free mice at 3 and 12 weeks old, so young and adult. And instead of colonizing it for 2 weeks, we colonize it for 1 week. And then we collected tissues and performed bacteria-specific qPCR. So in this case, basically, we identify SFB with qPCR. And we confirmed that both young and adult mice were properly colonized with SFB, as we can detect pretty nice levels of SFB in fecal pellets. But interestingly, we only find SFB DNA in the thymus and in the MLN of young mice, but not in adult mice. To understand if this process was selective for these two tissues, the thymus and the MLN, or if it was just spread all over the body of these mice, we colonized, again, B6 mice at weaning, and after 1 week of colonization, we collected multiple tissues and performed SFB-specific qPCR. And we found that this concentration of SFB DNA was primarily found in the thymus and MLN, but not in other tissues such as liver, spleen, heart, or lungs, suggesting that this was a directed process and it did not happen at random due to leaky gut or something similar. Interestingly, we found that there was a pretty strict time regulation of this process, since when we colonize mice at weaning with SFB, we found that 1, 2, and 3 weeks post-colonization, these mice remain colonized. However, SFB DNA can only be found in the thymus and MLN only 1 week after colonization, and this signal disappears promptly after 2 weeks or 3 weeks after colonization. To understand if this process was basically common for different bacteria, and not just specific for SFB, we also colonize our mice with E. coli, and we follow them for 1 week, and again, just as for SFB, we perform E. coli specific qPCR. Similarly to SFB, we find good colonization levels in fecal pellets, and just as with SFB, we find E. coli DNA only in the thymus and MLN of mice colonized at weaning, and not when they were colonized as adults. Good. In addition, we wanted to test if all bacteria were basically doing or getting into the thymus in a similar fashion. So we tested, instead of testing just SFB or E. coli specific DNA, we tested total 16S to assess for all bacteria going into the thymus. And of course, when we colonize with SFB or E. coli, we find this signal for total 16S in the thymus. But interestingly, in untouched mice, so basically B6 mice at 4 weeks of age that have not been colonized with anything, we find a pretty significant level of 16S DNA in the thymus, suggesting that this process may be used by multiple bacteria in the gut or perhaps other mucosa as well. So we collected the thymus of these untouched, untreated mice, and we performed 16S sequencing to see what bacteria were getting there. And interestingly, we found that in the thymus of untreated mice, so basically controlled B6 wild-type mice, we find a plethora of bacteria getting into the thymus. And interestingly, the composition of these bacteria resemble nicely to the one found in the cecum or in the MLN as well of these mice, mirroring whatever DNA bacterial composition we see in the intestine to the one we see getting into the thymus. And of course, to confirm that this process was again driven by the microbiota, we depleted the microbial colonizers in the gut with antibiotics at weaning, and we found that when we deplete gut microbiota with antibiotics, there's of course no colonization, as seen with no 16S DNA in the fecal pellets, and we completely erase the 16S signal in the thymus and MLN when we deplete the microbiota in the gut. Good. Now, since we know that we can find SFB-specific T cells in the thymus and also SFB and E. coli specific DNA in the thymus, the next question was how is this DNA getting there, and how are these SFB-specific T cells being expanded? Is there any APC from the gut that is traveling all the way to the thymus to perform all of these actions, or was something else happening? So we decided to analyze what APCs were changing in the thymus. So what we did was, again, colonized with either SFB or E. coli, young and adult mice for 2 weeks, and then we collected the thymus. What we first find is that in young mice, so mice colonized at weaning, there was an increase in total dendritic cells after SFB colonization or after E. coli colonization. However, in adult mice, we don't see any increase after colonization. Then, to understand exactly what dendritic cell subset was responsible of this increase, we look into the main two dendritic cell subsets from the gut, which are CX3CR1 positive dendritic cells and CD103 conventional dendritic cells. And here I'm showing mainly the CX3CR1 positive disease, and we see clearly that in frequencies and numbers, the CX3CR1 dendritic cells are the ones that are being enriched in young mice, but this enrichment doesn't happen in adult mice. And there is no, I'm not showing it here, but there's no change in CD103 dendritic cells in the thymus. To test what effect did depletion of gut microbes had on total dendritic cells in the thymus, we depleted the gut microbiota for 2 weeks with antibiotics given at weaning, and then we analyzed frequency and numbers of total dendritic cells, and we see a significant reduction, which actually was mainly triggered by a decrease in CX3CR1 positive cells. But in numbers, since all of the dendritic cells were going down, so were the CD103s and the CX3CR1 positive cells. To test if the CX3CR1 positive dendritic cells were actually the ones responsible of inducing these effects in the thymus, we colonized with SFB and with E. coli CX3 DTR mice. So basically, these are mice in which we can delete all the CX3 CR1 positive cells with diphtheria toxin injections. We did this for 1 week, and then we performed bacteria-specific qPCR in the thymus to assess for any changes. When we don't colonize the mice and we just assess for total 16S in the thymus, we can clearly see that depletion of the CX3CR1 positive cells actually significantly decreases the load of 16S DNA in the thymus. Similarly, when we colonize these mice with either SFB or E. coli, we can see a significant decrease in the bacterial DNA reaching the thymus. This suggests that the CX3CR1 positive cells are responsible for at least getting the DNA into the thymus. Similarly, we try to assess any change in the SFB-specific T cells being induced in the thymus. So we colonize with SFB these CX3 DTR mice, depleted the CX3s, and then after 2 weeks, we assess for SFB-specific T cells. And what we found is that depletion of these CX3CR1 positive cells actually decrease significantly the frequency and numbers of the SFB-specific T cells generated in the thymus, suggesting that these cells were required for this process. To understand if antigen presentation by these cells was also required for this process, we use our CX3 MHC knockout mice, in which by injection of tamoxifen, we are able to basically stop antigen presentation by inhibiting the expression of MHC class 2. So we colonize this mice with SFB, and we analyze SFB-specific T cells in the thymus. First, we realize that by blocking MHC in the CX3 positive cells, we are not decreasing the amount of SFB DNA found in the thymus. However, we are decreasing significantly the frequency and numbers of tetramer positive cells. So basically, our SFB-specific T cells are not being generated when we block CX3 cells of the ability to present antigen. Since we know that these CX3CR1 disease are important for the process of getting bacterial DNA into the thymus and important to induce bacteria-specific T cells in the thymus, we then wanted to know if these cells were actually migrating from the thymus. So for this, we first analyzed the expression of XCR1 and CERP alpha, which are common markers to basically assess if a cell or if an antigen presented cell is going to be mainly resident or if it's going to have migratory abilities. So for this, we just collected the thymus of mice at 5 weeks of age, and we compare these markers in total dendritic cells, in which we can see a split of resident and migratory APCs. However, basically, the majority of the CX3 CR1 dendritic cells were CERP alpha positive, suggesting that they had the migratory capacities, unlike the CD103 dendritic cells, which were mostly resident. And similarly to the CX3 CR1 dendritic cells, the plasmacytoid dendritic cells in the thymus were also and are known to migrate as well. So at least CX3 CR1 dendritic cells had the potential to be migratory cells. To actually test this migratory capacity, what we did was using KIGR mice. So basically, these mice constantly express green fluorescent protein in all their cells. However, when these cells are exposed to a 405 nanometer laser, these cells become red. So we can track which cells were actually exposed to this laser. And to use this in our migratory experiments, what we did was expose the cecum of these mice and only the cecum to this laser. Then we waited for 48 hours and we assessed where are the red cells that were supposed to be in the cecum. Where are they in these mice? Can they leave the cecum and go somewhere else, or were they all just remaining in the cecum and no migration happened? Interestingly, we find in both, in mice at 5 weeks of age and at 14 weeks of age, and we're choosing these time points because this is the time point, basically week 3 plus 2 or week 12 plus 2, in which we see these CX301s accumulating in the thymus. But we can see clearly that after shining the laser in the cecum, we see sickle cells, sickle red cells in the spleen, in the MLN, and in the thymus. And we can see clearly that the majority of these cells migrating from the cecum into these distal tissues were MHC class 2 positive. So basically these were antigen presenting cells. To dissect a little bit more what kind of cells were migrating from the cecum into the thymus, MLN, and spleen of these mice, we perform deeper flow cytometry characterization, and we realized that the majority of cells leaving the cecum and getting into the thymus were actually CX3CR1 dendritic cells. However, in the MLN and spleen, this population, the CX3CR1 dendritic cells, were not the majority. And interestingly, in adult mice, although we see similar levels of migration from the cecum, what we see is a completely different composition of cells that were leaving the cecum into these distal tissues. And the most striking difference happens actually in the thymus, where the CX3CR1 positive dendritic cell population, which was a majority in young mice, is no longer the majority and is decreasing as mice age. And in the MLN and in the spleen is mostly B cells that are leaving the cecum and reaching these sites. Then the next thing we wanted to do, since we see that these CX3CR1 positive dendritic cells are able to leave the gut and reach distal tissues such as the thymus, in which they are carrying bacterial DNA and inducing the production of microbiota-specific T cells, we wanted to figure out why is it so different between young mice and adult mice. So basically, what are these differences? Are there changes in the chemokine levels or expression in the thymus of young versus adult mice? Are the CX3CR1 cells different as mice age? So to figure this out, we collected the thymus of mice at weaning or at 12 weeks of age, so basically young versus adult, and we enrich for the epithelial part of the thymus. So basically, we got rid of most of the CD4 and the CD8 and the double positive cells, and we basically use most the enriched epithelial thymus to perform a chemokine analysis using qPCR. And what we find is that compared to young mice, adult mice had actually a decrease in CCL3, CCL5, and CX3, so 3X, C3L1. So basically, CCL3 and 5 are ligands for CCR5, and CX3L1 is a ligand for CX3CR1. So that was pretty interesting to us to find this difference in the chemokines that can be at least transcribed in the epithelial found in the thymus of young versus adult mice. We then analyzed any change in the CX3CR1 positive dendritic cells in the gut of young versus adult mice. And what we found is, after testing several chemokine receptors, is that the most significant decrease as mice age is a loss of the CCR5 positive CX3CR1 dendritic cells. And this correlates nicely with a loss of also the CCR5 ligands, CCL3 and CCL5. So since we see relevance or these changes between young and adult mice in CCR5 ligands and CX3CR1 ligands, we then decided to block all of these components in order to assess their role in migration of CX3's dendritic cells. And this was assessed by basically testing SFB specific qPCR in the thymus. So for this, we have regular wild-type mice, which are our CX3GFP+. We have CX3CR1 knockout mice. So basically, these mice, they have the CX3CR1 positive cells, dendritic cells. They are there, but they do not express CX3CR1. So basically, they cannot interact with the CX3CR1 ligand. We also, in both in wild type and in CX3 knockout, which is what we're calling it now, we block basically CCR5 ligands with TAC779, which is a CCR5 antagonist. However, since some of these ligands, mainly CCL2, which also targets CCR5, also targets CCR2, we decided to block CCR2 signaling by blocking its ligand with an anti-CCL2. So here we're going to show all the different combinations of these blocking CCR5, CCR2 signaling, and CX3CR1. So as I showed before, in mice that were not blocked of CCR5 or CCR2 and that were wild type for CX3CR1s, so basically wild type mice untreated and just colonized with SFB, we see pretty nice levels of SFB DNA in the thymus. However, in these wild type mice, when we block both CCR5 and CCR2 by administering TAC779, we see a significant reduction in the levels of SFB DNA. When we block only CCR2 with anti-CCL2, in wild type mice, we don't see this decrease at all, which suggests that CCR5 signaling is responsible for these effects. And when we block only CX3CR1 using the CX3 knockout mice, we see also a pretty considerable effect in a decrease of SFB DNA in the thymus. And when we block all, basically CCR5, CCR2, and in mice that do not express CX3CR1, we basically find no SFB DNA in the thymus of these mice, suggesting that mainly CCR5 and CX3CR1 signaling is important for the bacterial DNA to reach the thymus. Good. Now that we know exactly what is changing between young adult and adult mice, how this bacterial DNA and how these bacteria-specific T cells are generated in the thymus and which APC is responsible for these changes, then we wanted to know if these bacteria-specific T cells from the thymus, generated in the thymus, were actually functional. So we collected naive microbiota-specific CD4 T cells in the thymus and tried to assess their differentiation and their capacity to induce disease. So for this, we used the T cell transfer colitis model, in which we used donor mice, and these were our mice that were colonized with SFB, and I'm just showing here with SFB, but the same thing happens with E. coli, and with these mice were colonized with SFB at weaning, and then after 2 weeks, we isolated the thymus. We collected the thymus that had the SFB specific T cells, and we depleted CD8 T cells to basically have cell suspension that was enriched for CD4 single positive cells. And then we sorted naive CD4 positive T cells. Then, and we made sure that these cells were CD73 negative, and this is to help us that basically we were excluding all mature recirculating T cells from the periphery. And we transferred these thymic naive CD4 T cells into RAC knockout mice, our recipient mice. So basically these are mice that lack T cells and B cells and develop colitis when they receive naive T cells from a donor mouse. And after transfer, we're gonna follow these mice for a few weeks, and then we're gonna assess weight changes, colitis score, and basically analyze the immune cell landscape that happens after transfer. And just to be clear, our donors and our comparison is going to be mainly the donors, so basically mice that were not colonized with SFB versus mice that were colonized with SFB, and the recipients are going to be all colonized with SFB. So what we found was that recipient mice that received naive T cells from SFB colonized mice actually exhibited significant weight loss compared to control mice. The significant weight loss correlated nicely with an increased immune infiltration into the colon lamina propria, as we can see clearly here, compared to control mice, and this is 4 weeks after transfer, which I don't know, you may have done this process before, and usually it takes around 8 weeks, but basically this process was sped up with SFB specific T cells. And when we analyzed then the immune cells infiltrating into the colon lamina propria, we found that in the SFB, so basically recipient mice that receive cells from donors colonized with SFB versus control donors actually had these SFB specific T cells enriched in the colon lamina propria. To assess if CX3 dendritic cells were actually important for these effects, we repeated the same process, but in mice lacking CX3 cells, and we see clearly when we deplete from the donors the CX3 positive cells, we see much less infiltration and much less pathology, and of course, from this infiltration, we see much lower levels of SFB-specific T cells in the lamina propria. Similarly, when we repeat this in mice in which CX3 positive cells lack MHC class II, so basically mice that were not able to present the antigen to developing T cells, we see much less pathology and also much less SFB-specific T cells in the lamina propria. And I need to highlight that all of these SFB-specific T cells found in the lamina propria from the thymus of our donor mice were mainly Th17 and Th1/Th17 double positive cells. Good. So with this, we propose the following mechanism in which young mice, so mice at weaning, are first going to have their CX3 dendritic cells uptaking antigens in the gut. Then these cells are going to migrate with the bacterial antigen all the way through to the thymus. And in the thymus, they are going to present bacterial antigens into developing naive T cells, expanding the number of bacteria-specific CD4 positive T cells we find in the thymus. And then these cells are going to be exported into distal tissues, which could also be in the gut to exert their effects. However, in older mice, this process is stopped, which is much lower migration of CX3 dendritic cells, much less bacterial antigens in the thymus, lower or none, basically, SFB-specific T cells in the thymus, and well, basically nothing is coming out of the thymus. And when we deplete the microbiota with antibiotics at weaning, we basically see a stop of all of this process, suggesting that microbial colonization that happens spontaneously at weaning is driving all of these effects. And it's happening only when the microbiota is still developing, but stops in adult mice when the microbiota is fully established. So with this, you can understand we have plenty of questions. We have multiple feature directions that we can discuss after this talk. And with this, I want to close, and I want to thank everyone in my laboratory, which is the DL lab, which Dr. DL, which is super supportive and help us with all of these assays and planning and stuff. This project was basically also started with Professor Matt Bettini, who is at Utah right now. And this was possible with the help of Dawson Kim to help us with all the kick jar experiments, and with Kendra Norwood, who is no longer in the lab, but help us a lot with plenty of the mice work. And with this, I want to close. I want to thank everyone for listening to this talk. And if there are any questions, I would gladly answer them. Thank you very much.
Very nice material. Thank you, Dr. Zegarra-Ruiz. We very much appreciate it. There are some questions coming in, and I'm sure there'll be some more as well. Let me grab control of the screen back here. Just one second. Bear with me. The first question was from Kevin Tsai. How did the gut permeability of these mice change? Does colonization at different time points change permeability? There we go. Try that again. There, Dr. Zegarra-Ruiz, can you hear us?
Yes. Okay. Now I can hear you. Yeah.
Sorry. Yeah. Did you get that first question there?
Yes. Yes. I heard that. Okay. Yes. Perfect. That is something that we haven't tested yet. So we were planning to do different FITC assays at weaning and also in adult mice. Although we don't think that this is due to changes in permeability, because data that I cannot, I'm not showing it there, but we know that bacteria is basically uptaken by the CX3 positive dendritic cells in the gut, because we've tested basically died bacteria. And let me share my okay. Yes, so we've tested stained bacteria, and we know that this stained bacteria is only uptaken by CX3's dendritic cells like minutes after gavage, and is not free floating in the blood or anywhere else. So this has to be attached to some APC. So we don't think that is likely that is due to changes in permeability, but maybe in older mice, and I mean really older mice, like at least 2 years old or something like that, there could be an influence of impermeability, and that's something that we really want to test, because right now we're just showing data from mice at weaning and adult mice, some mice around 12 weeks of age, but it would be interesting to see changes in really old mice.
Great. And then we had a question from Minting Lee. In a typical situation, does SFB colonize murine gut until weaning or earlier?
So normally in any vivarium, if there's SFB, well, the breeders are going to be colonized with SFB, and the mice as well, the pups as well. So that is not something that you can time. So it's going to be present at any time point. In our facility, we are making sure that all of our mice, at least in one of the rooms, is SFB free. and that helps us to time the colonization whenever we want.
Okay, great. And then Lynn Lim asked, where in the thymus are you looking for 16S RNA? Is it total cell lysate?
Yes, this is total cell lysates. Ideally, we're going to be doing FISH in the future to know exactly where is this DNA getting into the thymus, because we all know that location is going to be critical to understand what is going to be the downstream process after antigen presentation. But we know for sure that these, at least, bacterial antigen presentation by CX3 DCs are not driving Tregs, at least for bacterial antigens. And we know this for SFB. However, we want to test this with different bacteria, like Clostridia and other Treg inducers, to know if this process is also not going to induce Tregs, or if it's going to do exactly the opposite.
Great. And we still have 6 or 7 minutes, so we're going to answer as many of these questions as we can, but if there's still several hundred people on the line, so if you have additional questions, keep them coming and we'll answer them either if we can get to them before the top of the hour, we will, and if not, we'll answer them in writing afterwards via email. So Damien Maceda asked, do you have evidence of those DCs detected in the thymus carrying any potential antigen from the transferred bacteria?
Yes, so that is one thing we're doing right now. We're doing sorting of CX3 CR1 disease in from the lamina propria, from the thymus, to know exactly if they are going to carry any specific bacterial DNA. And we're going to try to see if that's the case. But like I mentioned before, basically we know by dye and E. coli specifically that is the CX3CR1 positive disease, the migratory ones, the ones that are responsible for uptaking this bacteria and taking it away from the gut. But it's something that we're working on to really prove that this is happening only on CX3s at this time point, because like I showed before, this process changes as mice age as well. So there's a lot of work to continue these ideas.
Great. And then Lynn asked again, do you see any immune phenotype, for example, gut inflammation, if mice are ABX treated upon weaning?
We haven't tested that much more than what we're showing here, but we have done ABX treatment in really young mice at 4 and 5 weeks, and there's no phenotype, at least for gut inflammation, unless it's followed by DSS, then we see a slight pretty dramatical changes. But for antibiotics alone, we haven't studied that in much detail. However, we're also doing only a few weeks to a week of antibiotics, and then we just allow them to be recolonized. So I don't think it's going to have much long-term consequences unless the mice are really prone to developing gut inflammation, or if any other exogenous factor is added. However, that leads me to talk about how the importance of the host genotype is critical for this process, because all of this happens in wild-type mice, right? But we're now trying to elucidate if this process happens in mice prone to develop colitis, for example, in altered knockout mice, or in different strains of inflammation-prone mice as well.
Great. And still 3 or 4 minutes to go till the hour, so if you're okay, I'll keep going with the questions. Okay, super. This is from Jason asking if there's any significant depletion of CXC3CR1 positive DC in other sites, or is it just total affected of DTR depletion?
Yeah, so basically we're showing here changes in the thymus mainly, but we know that depletion of CX3CR1s with DT, at least in the CX3 DTR mice or the DL mice, is going to bring down the numbers in the colon lamina propria as well. So this is pretty much a general depletion of CX3 cells. That's why we went on and moved on with the studies with KGR to know specifically if these were the CX3s coming from the gut that are important, or if or not.
Okay, great. And then a question from Chloe Payette. Sorry if I mispronounced your name there. Hi, did you look if bacterial DNA induced inflammation in the thymus by inducing interferon or other pro-inflammatory molecules? And in which cells are located the bacteria DNA?
Yes, that is very important. We haven't tested any inflammation in the thymus by interferon or other IL-16F, any other pro-inflammatory cytokine. That is something that we look into doing, but it would be highly unlikely because we leave these mice age normally after colonization, and there's no overt phenotype other than what we show when we isolate these cells from the thymus into rat knockout mice. And to know specifically which cell has it, well, like I mentioned before, with the stain bacteria, we know it's at CX3 coming out of the gut that are carrying, primarily, these are the cells that are carrying this bacterial DNA. And yes, but we need to keep on exploring these ideas with sequencing and sorted cells to really confirm that it is these cells that are traveling through the blood and getting to the thymus carrying these this bacterial DNA.
Okay, and we got, I think, 2 minutes left. There's still over 200 people on the call, so it seems like people are interested in hearing these answers. So I'll keep going as long as you want to here. Damien had another question here. Can thymic stromal cells cross-present bacterial antigen brought into the thymus?
Yes, that is something really interesting that we really want to look into, because, of course, right now we're just showing that the ability of these CX3 dendritic cells to migrate outside the gut into the thymus and to express MHC class 2 to actually present the bacterial antigen are critical for the development of these tetramer-specific T cells, so the microbiota-specific T cells. But we cannot rule out that other APCs in the thymus are also mediating this process, or if there's some kind of crosstalk between these APCs. So that is something that we would like to explore in detail later on.
Okay, thank you for being a sport and answering so many of these. I think there's less than 10 questions left, so we'll just see what we can do here. Tran asking, great presentation. Have you checked any mature marker on the thymus epithelial cells of young mice? For example, FOXN1, PSMB11. If not, what is your hypothesis on maturation of thymus epithelials in CXCR knockout?
Well, we are not really looking into actual thymic epithelial cells. We were focusing mainly on CX3 since depletion with this cell, of these cells actually impair the phenotype that we find. But again, we cannot say that this is all driven by only CX3 disease, but at least at this time point and with the bacteria we've tested, these are the main players in driving these effects. But we can definitely keep on looking into other APCs in the thymus, resident or not.
Okay, and just as I said that, a whole bunch more questions came in, so we're definitely not going to make it to the end of them, but we'll get back to you in writing. I think we'll ask 2 more. How's that sound? This one came in from Ivan or Ivana. There we go, Ivana. How come these cells are positively and not negatively selected?
Yes, that is the big question. So we are really looking into it. We believe that it could be by signals driven from the gut environment that is allowing the CX3s to not drive negative selection of these cells, because negative selection is just not happening. These cells are being exported from the thymus. They are presenting all the markers related to positive selections. These are not dying; they are in fact proliferating, and they are active also outside in the periphery. So we need to know exactly why the CX3 positive disease have this ability. So that's why we're gonna do basically sequencing, RNA sequencing of CX3s in both thymus, lamina propria, in young, in adult mice, and also in different sites, to know what is so special about these cells, and also to know which signals are coming from the gut that is allowing these APCs to mediate positive selection. Although we're not really showing positive or negative selection, but the fact that they're proliferating and being exported of the thymus suggests that that's the case. So we are gonna keep on analyzing that to really find out what's happening, what is so special about this time point and about these cells that is allowing for expansion of microbiota-specific T cells in the thymus.
Okay, great. And the very last question for today, I think this is a good one, came in from Yuri Ritter. Maybe I missed it, but have you sorted or purified gut-derived thymus homing DCs after photoconversion for subsequent detection, verification of bacterial DNA or antigens?
Yes, no, that is also another thing. That is a good idea, actually. We haven't thought about that. But yeah, we can use the KGR mice to test for sorted RFP positive, MHC positive, CX3s. That would be the best way to actually focus on these cells that could be driving bacterial antigens. Also, I didn't show, because the KGR data that I show is from uncolonized mice, so basically mice with regular SPF microbiota. But it would be nice to show what happens when we deplete them with antibiotics, if all of this changes. And again, if we age them even more, to know if this process is also altered, and what happens in inflammation and other cases, right? Because there are many ways in which we can alter traffic of APCs from the gut into different organs, including the thymus, and inflammation or gut inflammation could be an important one to keep on studying.
Well, Dr. Zegarra-Ruiz, I really want to thank you for your time. I know we went a few minutes past the hour, but there were so many people still online and just questions continue to roll in. So I will be sure to email those all to you and to the people over at BioLegend as well. Thank you so much, BioLegend, for sponsoring these types of webinars. Obviously, there's great interest from the community and it brings fantastic content out there to researchers around the world. So BioLegend, thank you very much. Dr. Zegarra-Ruiz, thank you very much. And thank you everybody at home for attending at home and in your offices. It's been a fantastic webinar and wonderful participation, fantastic questions. Thanks everyone, and we'll see you again soon.
Thank you.